Hi everyone. I am Tanushree Gupta from Food System Integrity Team from New Zealand, and we are based in Palmerston North, which is on the North Island of New Zealand. So we have been working on dairy microbiology, meat microbiology, even water quality a lot. But having a bit of a background on that side, we have also started working on some natural antimicrobial production from bacteria. Now, I was invited to talk about one of the review papers my PhD student published, and it was specifically on the antimicrobial production by anaerobes, and particularly the Clostridium species. When we talk about different antimicrobials or natural antimicrobial compounds, we always have in our mind that what is actually the need for it, right? I'll just move this one here. Sorry. Um, so we know that antibiotic resistance is increasing tremendously, and somewhere we have to, I'm not saying blame, but global warming or climate change has a lot to do with it, right? So, talking about antimicrobial resistance, talking about the evolution of bacteria each decade, each minute, that all contributes for a need of a new compound, whether it is synthetic or natural. Okay, so with synthetic, what happens is obviously now we are moving towards more eco friendly compounds. And with the synthetic ones, there are a lot more resistance within microorganism. And again, it goes back to global warming, which um, integrates more pathogenicity from either virus, as we know with COVID cases we are having at the moment. Not even that, but bacteria evolved, fungus evolved. So all of these have their opportunity to come up and evolve during this time. So when we talk about different diseases, not only human, but it may affect plants. And then what next? Synthetic, people are going away from synthetic compounds. So there is more and more need of natural compounds because we believe that it may fight antibiotic resistance. And then coming from New Zealand, even though we have lot more rain, lot more winter season, but if you look into the differences between the two sides, we are estimating that the 2021 summer, which has already started, till March or April 2022, there will be a huge difference between the temperature, and we are expecting more droughts, more higher temperature all across New Zealand. So that might shift or that might change the need or upcoming of the diseases, and we should be prepared with it. So, as part of that, we have come into a colloquium that we need more natural compounds. So, why not start looking at other bacteria, specifically anaerobic bacteria? So, if you all are aware, a lot of work has been done on identifying and characterization of bacterial antimicrobials. So, and a lot to be connected to streptomyces and the most famous lactic acid bacteria. However, anaerobic bacteria has been largely neglected, mostly because their growth conditions is highly challenging. So you have to have fermenters, you have to have proper growth conditions so that you can grow those anaerobes and get the antimicrobial compounds out. Not only that, but it has also been associated with some negative imaging, specifically with Clostridium species, that they cause and they produce a lot of toxins, for example, perfringens, Clostridium botulinum, or difficile. Yes, there are, but a lot of percentage of Clostridium species are actually saprophytes, and they live within our gut. They live happily, friendly way in our environment. So it most of or the maximum of these species don't actually cause any disease, or they don't produce that much of toxins. So why that has been that it is largely neglected. Now if you look into some studies, and I have quoted only two at the moment. A lot of work was done in 1960s or 1970s where they found or isolated and detected bacteriocins from non-pathogenic Clostridium species. Okay. But after that, they stopped. So the maximum you'll get is from 1980s, and after that, they have stopped working on anaerobes. Now, it is duly just because it was highly challenging to grow them and extract the antimicrobial component, or just they wanted to move out, because they might have found, or the researchers might have found that bacillus was easier to grow and they were spore forming bacteria as well, then why not work more on that? Um, so, late 2000s or in the 2000s era, the whole computational genomics or genetic approaches came into light. And with the advent of those technologies, people started working, or the researchers started working more and finding more antimicrobials from Clostridium botulinum, for instance, and some others like Clostridium cellulolyticum, cellulovorans. And just because on the basis of the upcoming computational tools and the lab adapted growth conditions, we were able to generate a manuscript, a review in which we could gather some of the important information and show that Clostridium could be a viable source of antimicrobials. And not to forget, lately it has come to our notice that Clostridium species, specifically Clostridium butyricum, can also be used as probiotics. And it has been seen to increase the immunity and growth performance in fish. Tilapia, in broilers, they have increased digestive function, growth performance has been increased, immunity has been increased in chickens. And not only that, it has also increased the gut function of human beings as well. So this Miaricin are the tablets which are from Clostridium butyricum and is available in market, and that has been used by human beings for a good gut system. And also it has been found that Clostridium butyricum miari 588, which is actually linked to these tablets, they are associated as anti-cancerous agents as well. So obviously, looking into so much of data, we think and we hypothesize that there is a lot to still look into Clostridium species and see what they can produce. So far we have known that a lot could be done in lab, and how you can modulate the culture-based methodologies to enhance or to identify antimicrobial compounds from Clostridium species. So the benefit of using Clostridium species on top of that, they are spore formers, and you can actually work as such with the spores. You don't have to rely on getting the vegetative cells. So yes, they are anaerobes, but most of the Clostridium species can grow in 10% oxygen as well. So you can very well utilize those growth conditions to explore or to detect some antimicrobials. So what I was saying, that the most important bit about Clostridium to look into is that they survive, as compared to other environmental bacteria and bacillus spore forming species. They can actually survive in environmental conditions way better than other bacteria. And it is just because they have got a variety of metabolic pathways, because they have to switch on different metabolic pathways when they are in a farm environment, when they are in soil, when they are in gut. So they can actually modulate their metabolic pathways. So that indicates that if they can modulate their metabolic pathways, there are more chances that they can switch on and off their variety of biochemical pathways within themselves, and that might switch on some of the pathways that may produce antimicrobials. Now, when it comes down to culture based methods in the lab, it is highly challenging to cope or to get what the environmental conditions are for thriving these bacteria. But some research has found that you can actually add a few bits to your lab conditions to enhance the production of antimicrobials. And one such good example is actually you can see on the bottom of your right hand side that when they were giving the standard fermentation culture conditions to Clostridium cellulolyticum, there wasn't any bacteria in production. So this closthiamide is one of the antimicrobial compounds, which was supposed to be produced from this organism, but it wasn't. And they also tried different nutrition stress, different antibiotic, temperature, pH stress, and they were looking for if there could be a possibility of producing clostridium, but actually they couldn't. But what happened? They got an aqueous soil mixture. They sterilized it, and then when they added into the culture condition to the Clostridium cellulolyticum, boom, there was this production of clostridium. So, and not only that, people have also shown that if they added fecal material or just a solution of human feces in water and added it to the culture conditions, the sporulation of Clostridium species, and this is particularly with Clostridium cellulolyticum again, the sporulation increased, enhanced like around 300 times, and it also downregulated its motility as well as toxin production. Also, they have shown that if there is an antibiotic treatment for C. difficile in your gut, it utilizes succinate to produce butyrate, which actually aided its growth, or it enhanced the population of Clostridium difficile in gut. So that's why it is suggested that you have to be very careful that for how long you are given antibiotic treatment for, because you might be treating some other disease or other medical condition or bacterial condition, but actually you are enhancing the population of Clostridium difficile. And then not only the antibiotic treatment, but it is also shown that it produces paracrisol, which is a bacteriostatic compound, and it enhances its survivability of Clostridium difficile over E. coli, other bacteroids, as well as Klebsiella oxytoca. So, as I mentioned earlier, in late 2000, we have seen a boom of antimicrobial compound discoveries from Clostridium species. So three major ones are closthiamide, which was the first ever antimicrobial, which was detected in, and it is part of polythiamide class, and it has been detected from Clostridium cellulolyticum, and it was active against methicillin-resistant Staph aureus, as well as vancomycin-resistant enterococci strains. It was active against a wide range of gram-positive bacteria, but not so much against E. coli, right? The next one is the clostridium, which was the first polyketide being found from Clostridium difficile. And it was effective against, again, MRSA, vancomycin-resistant enterococcus, as well as mycobacterium. Similarly, clostridioidan was also um, a detected and identified from Clostridium boehringer, which was also reported as mycobacterial. Then, as you um, as you shifted to more of the late 2000s, there was a huge range of different tools for genome mining, which actually threw a lot of focus and light on some of the gene clusters, which are known as biosynthetic gene clusters, and they are related to the identification. I wouldn't say the production at the moment. It is just the identification or detection of different ribosomal synthesized post translationally modified peptides, polyketides, non-ribosomal peptides like different antibiotics and bacitracins, these all, and niacin, these all come under these categories. Um, they have been identified. So the gene clusters for these particular antimicrobial compounds were identified using genome mining tools. And this has actually elaborated that how you can identify and differentiate between environmental strains to the human disease or human related strains, and they have seen a huge difference between the same species of clostridium, but each strain might have a different set of biosynthetic gene clusters for RIPPS or polyketides or non-ribosomal peptides. So, what these genome tools do, they take the whole genome sequence from each bacteria or each species. Then you use, or you can either use online tool like Anti-Smash or Art, or you can very well use some of the um, designed tools which you need to download it from the internet. It is not freely available, but you definitely need a bioinformatician along with you to deduce what type of gene clusters you can identify. So you take the whole genome from your microorganisms data, then you put through that genome, the whole genome sequence, to those online or specific tools, and then you can straight away see that which gene clusters you can actually look for. And this particular FSO1, FS2.2, these are actually our isolates, and we actually found a range of bacteriocin, a range of ribosomally synthesized post translationally modified peptides, SACT peptides, which come under RIPPS, and a couple of them had polyketide gene cluster. Now, they do identify the gene clusters, but we haven't gone that far that that these tools can actually tell you whether they produce something. It is just because not many people have been working on it, so the database to which these tools consign or identify which cluster it might be associated with, because the data is not so fully enriched at the moment. They can only link to the present RIPPS examples or polyketide examples, whether it's a bacteriocin or whether it's a polyketide. So it might end up, so if you put your sequence into it, they might tell you that it is 30% similar to such and such or 40% similar, but those percentage are really low. So it might indicate that there is a possibility that it is a new compound that you are looking for. So there are still a lot of gaps which needs to be filled in, right? Um, so, on the basis of these genome tools, we found, containing all the different reviews and literature survey, we found that there is a possibility that approximately 75% of the Clostridium species are actually capable of producing PKS, like polyketides or non-ribosomal peptides. But it all comes down to that how we can identify those. Okay, so as I mentioned earlier, so we have a huge range of gene clusters, but we can't really be sure that those gene clusters encode for the products, because those genes could be silent or could be downregulated. So we need to come up with a strategy which might induce the expression of those gene clusters to produce those products or antimicrobial. And one big example is what previously I have just mentioned, that you need to give some inducers so that they start producing those antimicrobial compounds. And when I was talking about the differences in the species, if you can see this correct nicely, you see you do have Clostridium, the same species, but the strains are different, but they produce different RIPPS products. One is lengthy peptide, the another one could be something else. Similarly, if you look down on your left hand side, perfringens, for instance, that kind one strain could be producing lesopeptide, the another one can be producing lactococin, which is other type of RIPP. And then another huge range of different antimicrobial gene clusters you can actually identify from Clostridium species. So moving forward, so we did do a quick search and saw that the ones what we see from Clostridium, is it very similar? Those gene clusters, are those very similar to other known gene clusters or biosynthetic gene clusters from known bacteria, such as Bacillus or lactic acid bacteria? So we used a online tool which is called Anti-Smash, and it is freely available. You can use it for fungi, you can use it for any other bacterial um, species and have a big search. So, what we found, and we took an example of Clostridium cellulovorans 743b genome, and we targeted lengthy peptide biosynthetic gene clusters, and we saw that it sat really uniquely around Clostridium cellulovorans, and it wasn't that that protein or the peptide composition wasn't really anywhere closer to Bacillus species. And if you look into this tree, into this map, you won't see lactic acid bacteria anywhere in this. So it was very much specifically, I would say, to a spore forming cluster. But again, within a spore forming cluster, it was way away from the Bacillus cereus group, but it was very much closely related to Clostridium species, and that too very much unique to cellulovorans. So there could be a possibility that these gene clusters are very specific to strain, very specific to species, but then on the other hand, they could be a very novel type of antimicrobials gene clusters. So we have to look into more in details, and whosoever works around it have to be very careful what they are dealing with. So there are certain challenges, but then we have opportunities as well with the advent of whole genome sequencing. The challenges are that we really don't know that what these biosynthetic gene cluster might be translating into. Right? What compound? Because as I mentioned earlier, we have limited resource, we have limited knowledge around what's in the database. So we have to do more and more studies so that we keep filling up that database, and people can use that resource or that reservoir to analyze their data and see what's the similarity is. Okay, so that's the main challenge. And then again, that the existing database doesn't have much information around Clostridium species. So what we need to do is we need to generate more information using genome mining around potential biosynthetic gene cluster, and need to prioritize on the known ones or on the biosynthetic gene clusters which codes for certain antimicrobial compounds, so that when we start doing some of our experimental designs, that it reduces our time and cost. Okay, then tools such as arts, apart from using anti-smash, you can use antibiotic resistance target seeker, CDD prism, that can give you a prediction of what secondary metabolites you might be detecting. CDD is it's really good, and it understands the chemical structure which could be associated with the product. So you may have a little bit of knowledge or an idea that what kind of structure of the antimicrobial compound you may end up getting, and then you can do a quick search around that, what homolog or what analog you can find around that structure, and it could indicate what antimicrobial product you might be getting. So, so what at the end I would say that if you combine the genomics and the culture-based approach, then you can very well understand the whole pathway and see that what antimicrobial compound you might be getting. So this on the gray side is the simple conventional approach you'll be taking, isolating the cultures from the environment, screen for what antimicrobial compound you might be getting using some screening tools, and seeing what which bacteria do they kill, and then you isolate, and you can introduce metabolomics here or proteomics here. You can isolate that compounds and then do more of a serious search and see what um, chemical structure those compounds have, and then look for putative antimicrobials. On the other hand, you can actually use the genomic data. You can use online tools to see what genome or what gene clusters you may get, and then what it might be looking into. And once you know that you have an idea that this is coming out to be similar to such and such, which is already present in the database, then you can tweak your environmental conditions in the lab or in your lab conditions, and then start screening and isolating those compounds as mentioned in the conventional approach. At the end, I would like to conclude that yes, there are difficulties, are challenges to look into novel antimicrobial compounds, but Clostridium, because they have a variety of places that or they can survive, so they have evolved their microbial or metabolic pathway, so they are a good source of looking into natural antimicrobial compound synthesis. So a lot more research needs to be done. There are limited research on the indication of putative antimicrobial peptide and secondary metabolite from Clostridium species, but with the advent of genome mining tools and genomics knowledge, in combination with some improved and modified culture methods, we may explore more and more antimicrobial compounds from Clostridium species, and then incorporation of those ecological niche in the laboratory system, we might enhance or induce the silent chain, which may again improve the bioactive or antimicrobial synthesis in anaerobic bacteria. Then, more importantly, we need to be very specific about the molecular approaches, what we are using. And you can also use modeling, epigenetic remodeling, or heterologous expression, or more and easily that would be the activation of the pathway of those biosynthetic gene clusters along with optimized growth conditions, which can promote the expression of those biosynthetic gene clusters to promote or to produce antimicrobial compounds. At the end, I would like to acknowledge and thank the International Conference on Microbiology Infectious Diseases to invite me to present this work, what we have been doing. And a very big thanks to Amila, who is my PhD student and who has moved to Aarhus University to do postdoc. And this whole anaerobic bacteria and antimicrobial extraction from Clostridium was his PhD. Kwang, who has worked a lot around this work doing antimicrobial screening tests, Dr. Gail Brightwell, who is our team leader, Professor Liang Li, who did all the metabolomics and is in University of Alberta, and of course our Ag Research Fund, who has funded this um, study. Thank you. Any questions? I can see someone raise their hand. Yeah, can you please talk?
Yeah. Hi, Tanushree. This is Vishwambar from India.
Hi.
So, yeah. So, as you mentioned, this Clostridium, it mostly acting against the gram-positive pathogens, right? And very partially acting against the gram-negative. So, based on your experience, what will be the reason behind this? It more acting against the gram-positive?
So what has happened is this was I have given you an example of only a particular um, what do you say, antimicrobial compounds at the moment, clostridiumide or clostribin and clostrindonil, right? So
Yeah.
it could be that it depends, it's the activity, the mode of activity is different, right? And because we know that gram negative may be an easy target because even though they have a complex cell membrane and cell wall, but it is easier to target gram positive because their lipid and the protein combination is very different. But I will give you a heads up. The study what we have done so far as part of my PhD students PhD, which I haven't showed it here because I was supposed to present the review paper we have got, but we have found some antimicrobial compounds which were equally active against gram negative. So it is just depends on what antimicrobials you are looking for, but there are a lot more other new compounds and some of the remaining ones which have never been targeted earlier, but they are equally active against bacteria, all sort of bacteria. But I think it has to do with the cell membrane. We haven't looked into because we didn't work on clostridiumide, and they haven't actually worked on that what was the mode of action. They just showed that this is coming out for the first time from Clostridium, cellular liticum, for instance, for closthiomide. But no, what was actually the activity, where it was actually acting, wasn't found. Yes, Martha.
Yes, you were describing the use of Clostridium in cancer. Have you, which types of cancer are more susceptible to this type of treatment?
No, we haven't. Actually, it wasn't
Oh,
us who have worked. It is a
okay.
part of literature search.
Literature search. Okay.
Yes, yes. But if I look into it, actually, they were anti-tumorous and they enhanced neutrophils through
Okay,
MMP8
enhanced
pathway.
neutrophils.
Yeah.
And what was that when they took it by mouth?
Which one? Those
This
ones.
one, the one where they advanced the neutrophils. So was that
Uh,
IV injections, part of like what they call a cobalt
no,
therapy,
no,
or
no.
that is just by ingestion?
This was, I think, just on the cellular level. So they did an in vitro cell line studies.
Okay, great. Thank you so
Yes.
much. Thank
Yeah.
you.
So these studies actually, until and unless they have actually worked on specifically animal models like broilers or fish or other bits, they haven't actually worked on against the anti-cancerous. They haven't worked on patient as such. On patient, they have only used these tablets, the Miaricin tablets, and they have actually shown that in some of the patient it has enhanced the gut microbiota and gut health.
Great. Thank you so much.
Yeah, no worries.
Yeah,
um
yeah,
yeah.
yeah. Tanushi, one more question.
Yes,
Uh,
please.
Yeah, particularly this, if you see these organisms, mostly these bacteriocins are the responsible for antimicrobial activity, right? And
Yes.
apart from this bacteriocin, if we also actually we are also working on the lactic acid bacteria. So
Mm-hmm.
we got the bacteriocin also and some other metabolites also, and
Mm-hmm.
even the peptides, small peptides also they are acting a good as a antimicrobial agents.
Mm-hmm.
But if you see compared to bacteriocins, like these metabolite, their purification and characterization is like a tricky job. So, can you suggest something to purify and characterize these secondary metabolites produced by this bacteria?
Well, because our work, I would say that overall, our all over the work we have done, we have looked for metabolites. And actually, metabolomics is not a tricky bit. It is easier than looking for the small peptides, because the small peptides, it's really challenging to actually bind to the columns. So I would suggest, if you're looking for bacteriocins, well and good, because you have got established protocols to work with bacteriocins. But with metabolites, have you worked with peptides so far? Have you looked
Not yet,
into
not
peptides?
yet,
Not yet.
not yet.
Okay, so with metabolomics, you can actually use the similar LCMS, liquid chromatograph, along with mass spec. So you can actually phase out. You can use different columns, and it is equipped, well equipped now. You can send your whole extract to different service provider, and they can fractionate out. So fractionation using LCMS has become really easy, and you can get individual fraction and then test those metabolites around, looking into which one gives you the antimicrobial properties. So it is easy. It's not a tricky job now. You can use LCMS very easily, and that's what we have done.
Okay, and see, suppose some like novel antimicrobial agents are there, those are not reported in some library or
Yeah.
anything. So that
Yes.
how will identify or characterize that molecule?
Yes, so that area that still needs to be developed a lot, because you need a combination of LCMS, but of a high quality. Along with it, you need NMR. which a lot of people don't have. And you need to know, using the genome tools, that what putative indication you are actually getting, that it could be a possibility that it is indicating that it is either a bacteriocin or it is a RPS or polyketide, and then you give different shots. It is challenging, I have to say, because we think with our Clostridium species, we have got a huge range of novel compounds, but we are still struggling to get the best shot with X-ray crystallography, the CDT to work, that what chemical compound structure we
Yeah.
might get, and along with it, NMR.
Yeah,
So,
definitely.
yeah,
Like the same things we are now currently we are following. We are purifying through this silica column, and
yeah.
then we are giving for NMR, XRD, LC, HRMS. So same problem we are facing, but this separation of this compound is a really tricky job, because we are sometimes we are getting the mixture of compounds. So it's NMR, you are not getting the pure peaks in NMR. So that's why we are sticking here. So
So you are saying that you're not getting from NMR. Is it the novel compound you're talking about, or is it the secondary metabolites?
yeah, it is a novel compound, but
Yeah, yeah.
yeah, but yet we have
Yes.
to characterize completely.
Yes, I totally understand. So are you specifically working? Oh, no, you said you were working with lactic acid bacteria. Now, that's the other thing, because people don't work on the novel compounds. So you really can't get anything because the data is really restricted. So the database, until and unless database is not enriched, people are not putting their compounds in there. It would be really tricky. It
Right.
is possible, but it has to be done with lots of combination and permutations, unfortunately, at this point.
Yeah. Yeah. Okay, thanks for your suggestions. We'll see the and in
Yes,
future
yeah.
also we are looking for like your area and our area is like similar kind of. We are working on the natural products. So in future, if we need some help, so
Definitely, yes, you can contact me. We can, I think you can get my email address from Isabel,
yeah.
who has been organizing this
Yeah,
whole
yeah,
thing.
I got it. You wanna we have one communication about this conference with this mail ID. So okay, whenever
Yes, yes, you can be in touch. Yes, yep,
yeah,
and
okay,
we'll be happy to help you.
yeah,
Yep.
thank you very much. Yeah,
No, thank you. Thank you so much. So it's
Yeah,
all good to stop presenting?
yeah, it's fine. And thank you for your patience and your valuable suggestions to the budding researchers in India. And we have one more final lecture for the day from Dr. Jamie from United States. Once we have done that lecture for the day, we'll close that. And you can expect your certificates by tomorrow end of the day.
All right. Thank you so much. Thank you, everyone.